Yeah, it's supposed to be
Can you name
one warm layer. If you're in a leg, warm
He
layer
put you in
from
the
the
email
bottom.
that when I was the entire department.
So,
I was like, oh, like 200 people
So-
just watched this.
So then we can use warm.
I'm gonna take notes. I do have to read.
Test you on it.
yeah. So it's just supposed to be
Yeah.
like enough cold for room temperature. Yeah. It's just
There's so
supposed
many
to
classes
be
now that are required.
really
you
low
to
room
go to
temperature
talks.
and then a layer of top.
I just want to write a
So-
flavor on it.
Today it didn't, but I don't know which way they did it. So it could be that if you have room temperature in your form, that I can call it the room temperature. And it's probably not
Like
that
regular
early.
spots.
Oh, there's some.
So nothing patched at all.
No, they did, but the other stuff is kept circulating for an entire time, which would be if you had a, so somewhat weird.
Do we have a sign in or you just put a?
What?
Do we have a sign in?
What I do is that I just look around and I put little X marks.
I found
So
the
when
paper.
it's here, X mark. When it's here, X mark.
Did you like that?
I
Yeah.
found the paper. It's for
And they said,
And also,
yeah, but it just, they said they put it in.
Thursday I do
like,
a class from 9 to 11, so I can finish early.
Yeah, I don't know, I'll,
With the online.
But if I came in at 11,
yeah.
I can go at 11:30.
I had to like look through something
Okay.
when I got like
It's
the
still-
whole
I just have
I
login
missed
one.
that that's like divided by this, but I'm just like, there's still gonna be this.
Yeah, we got everybody.
I can't figure out how to, it's like, you know how it's all like 15 years to now it's stream.
It's actually not roasting with this class right now. Yeah.
Because every time I try to like that, it's actually like valid.
They've been testing for like,
Look up there.
at this point in an hour, I don't know how good it was, and they weren't dense, it's like, it's good one.
Maybe I know like.
You know, in state school terms that are like, I'm graduating. I graduated. Look at me. And I'm like, still have over.
But yeah, mid.
Go to work for it.
It's super, super, super late.
You're bored.
It's like the middle. It's like not even the beginning.
You ever have to do that?
My kids never saw this. They died. Gotta come.
Ask Aaron.
Yeah.
You're looking up.
Oh, really? So after this is done, I'll just copy it. Did you send the, is it still like from last year, is it a new thing?
Oh, there's a new link, updated website there.
Can you send me?
Yeah.
Huh? Oh, no, I'm starting this part of it. Hello and welcome to the Chemistry Biochemistry Senior Experience. So I want to make a couple of announcements before we get to our speaker. The chemistry picnic is May 19th at 5 p.m. and that you do want to sign up outside the stock room downstairs so that we have a head count and make sure we know how many cows versus how many plants. Okay? So chemistry picnic, May 19th. Also, this is the very first of 16 presentations. And so our very own Sam Knott will be talking next Tuesday in this very same back channel and back place. Okay, so 11:10, and you can hear about transcriptional control and listeria. We also have Wednesday, Thursday, and Friday afternoons in the same room, a bunch of talks, which are all available. You can see the whens, whats, hows, and whos on the university calendar. Okay? If you happen to miss one, other than this class, you can get them either on YouTube or webcast. Link available also on the website. Are there any questions? So, without further ado, I'd like to introduce Andrea Wilkinson. I've known her most actually from basketball, more than anything else. So I've been her basketball statistician for the last 4 years. And so it's been an honor, and I don't do volleyball stats, so I haven't been a basketball statistician for her for 3 years. But she hails from Racine, Wisconsin, and is headed off to pharmacy school in beautiful Belmont, in Nashville, Tennessee, which my sister played basketball in Nashville. It's very nice. So anyhow, without further ado, I will let Andrea give her talk.
All right, thank you, Dr. Dave. Like you mentioned, my name is Andrea Wilkinson, and I'm here to present to you guys the research that I've been doing with Professor Judith Humphreys. The title of my project is Nuclear Translocation of the Transcription Factor NFkB in the snail Bienfilari glabrata. So to start with, I just want to give some background as to why this research is important, starting out with the significance of Bienfilari glabrata itself. So why even study this snail? Well, to start with, it is an air-breathing freshwater snail, and it is significant because it is an intermediate host for the parasitic worm Schistosoma mantsoni. And Schistosoma mantsoni does cause intestinal schistosomiasis in humans, and this is the most widespread version of schistosomiasis found worldwide, which is why this is important. So what exactly is schistosomiasis? Well, it's a parasitic disease caused by various species of schistosome worms. It's the second most common parasitic infection after malaria, and it infects around 240 million people worldwide in about 78 countries. And looking at the map here, we can see the distribution of where those infections occur. There is both intestinal and urinary schistosomiasis. So the red images here is going to be where both of those infections have been reported, and then the purple is going to be where only intestinal schistosomiasis occurs, and then the yellow being where only urinary schistosomiasis occurs. So not only is it the second most common parasitic infection, but it's also the most deadly neglected tropical disease, killing around 280,000 people per year. And it is interesting to note that there are 5 major schistosome species that do infect humans, including S. mantsoni, S. intercalatum, S. japonicum, S. mccongi, and S. hematobium. And each of these schistosome species do have their own specific intermediate host snail species, which you can see in parentheses here. Of these 5, the top 4 do cause intestinal schistosomiasis, while S. hematobium causes the urinary schistosomiasis. But then, like I mentioned in the last slide, my project focuses on the Schistosoma mantsoni and Bienfilari glabrata model here. So to better understand the role that Bienfilari glabrata plays in this disease, we can look at the life cycle of the schistosome worms itself. So the life cycle starts when the eggs are passed from the host in the feces or the urine. Once those eggs reach a freshwater source, they then hatch into myristidia. These myristidia are then free swimming and are able to penetrate into their specific intermediate host snail species. And then once in the snail, they develop into 2 generations of sporocysts. And as they exit the snail, they then develop into these cercariae. So these cercariae are what are infectious to humans. They're able to, once again in the freshwater source, able to then penetrate into the host snail skin. And during penetration, they then lose their tails, developing into these schistosomula here. Those schistosomula then migrate into the liver, where they're able to mature into these adult worms. There are both male and female adult worms, so once those adult worms pair together, they migrate into the intestinal veins, where they are able to lay eggs and start the cycle all over again. So looking at this, it's pretty evident that the snail plays a major role in keeping the parasite alive and continuing on its life cycle. However, it's interesting to note that there are 2 different strains of Bienfilari glabrata. So starting, we have the BS90 strain. This strain is resistant to parasitic infection, and then we also have the NMRI strain. So the NMRI strain is susceptible to parasitic infection. What this means is that when those myristidia penetrate the BS90 strain, they're able to fight off the parasite and kill the parasite, preventing it from continuing its life cycle. However, when the NMRI strain gets infected with those myristidia, they're not able to fight it off as like the BS90 strain. Therefore, the parasite is able to develop and continue its life cycle and be infectious to humans. So what exactly makes these two strains different? Well, in order to determine that, we can look at their immune responses. So biofilarial glabrata do have a variety of defense strategies, including phagocytosis, encapsulation of pathogens, secretion of lectins, and production of oxygen radicals. I'm not going to go and explain what each of these defense strategies are, but what this indicates is that they must be dependent on regulation of immune-related genes at the transcriptional level within the snail. So what exactly do I mean by that? Well, we can look at what transcription is. So transcription is generally where gene expression is regulated the most. So genes that can either be off or on, and in order to turn those genes on, they must get transcribed from the DNA into RNA, and then from there translated into these proteins. So the proteins are what is actually active in the immune response, and can therefore then function to get rid of those infections. So gene regulation can be a very extensive process, and there's multiple ways to regulate gene expression. So my project zooms in on that and focuses on NFkB, which is a transcription factor. So a transcription factor is a protein that binds DNA, and then it regulates gene expression by promoting or suppressing transcription. So as we can see on the diagram over here, NFkB is generally found in the cytoplasm, bound to this inhibitor protein called IkB. However, once IkB gets degraded, it releases NFkB, so then NFkB is able to function as a transcription factor, moving into the nucleus and binding DNA. So once it binds that DNA, it initiates that transcription, turning those genes on in order to create those proteins that can function in the immune response. So NFkB is significant because it is found in almost all multicellular organisms, and it's also important because it regulates gene expression for both innate and adaptive immune responses. So we ourselves as vertebrates do have both of these immune responses, the innate and the adaptive. However, invertebrates such as biofilarial glabrata or the snails only have the innate immune response. So I will briefly go over both of these, starting with the adaptive immune response. This immune response is only found in vertebrates, and it functions by creating immunological memory after an initial response to a specific pathogen. And this is largely done through the process of antigens and antibodies. So looking at how that kind of works, we can look at this diagram here. So the main cells that are active in the adaptive immune response are going to be these B cells, and they get activated once an antigen binds to that B cell. So the antigen is going to be any sort of foreign particle found within the body, such as bacterial particles, parasitic particles, and any other toxins. So anything that your body doesn't recognize as normal. So once that binds to the B cell, it's then signals to the B cell to replicate. So we can see in step 2 that it is dividing into multiple B cells. From there, it then is going to differentiate into these plasma cells and memory B cells. So the memory B cells here is going to have that specific receptor that the antigen initially bound to. And so this memory cell is what allows the adaptive immune response to remember that infection and can therefore fight it off more easily in the future if you're ever exposed to it again. And then these plasma cells are what is actually going to fight off that infection, and it does so by producing these antibodies. So the antibodies will then are specific to the antigen that initially bound to that B cell in the beginning, and then go ahead and bind to that antigen to get rid of those particles, those toxins within the body. And the most classic example of this is going to be vaccines. So when you get a vaccine, you are inserting those antigens into your body. So for example, if you get a measles vaccine, you're going to be inserting the antigens from the measles infection into your body so that your body can create these memory cells that are able to fight off that infection if you're ever to be exposed to it again in the future. So on the reverse side, we have the innate immune response. So this is going to be the primary immune response, or the only immune response found within biofilarial glabrata. And the main part I'm going to focus on within the innate immune response is going to be pattern recognition receptors and PAMPs. So the pattern recognition receptor is going to be receptors found on the surface of the cells, And then these are going to be specific to these PAMPs, or pathogen-associated molecular patterns. So these molecular patterns, once again, are then specific to that receptor, and once that binding occurs, it initiates this innate immune response. And some examples of PAMPs include peptidoglycan or lipopolysaccharide, or LPS. And these are representative of gram-positive bacteria or gram-negative bacteria. So peptidoglycan is for the gram-positive bacteria, which we can see here. So the cell wall of a gram-positive bacteria is largely made up of peptidoglycan, which you can see from all the red squiggly lines here. However, gram-negative bacteria have very little peptidoglycan in their cell wall, and instead have these LPS molecules sticking out from their surface. And so this LPS, or the peptidoglycan, are going to be that specific molecular pattern that is binding to this pattern recognition receptor within the organism, setting off that immune response. So in order to understand how that binding eventually leads to a response, we can look at what signal transduction is. So signal transduction is the transmission of molecular signals from a cell's exterior to its interior. So we can use peptidoglycan as an example here. So the hormone or environmental stimulus, this would be the peptidoglycan molecule, and that is then going to bind to the pattern recognition receptor that we see on the surface of the cell. Once that binding occurs, it causes this transduction or relay proteins and second messengers. So essentially like a signal cascade that then ultimately leads to activation of a cellular response, or in this case, an immune response. My project specifically looks at the signal transduction involving NFkB, or the NFkB pathway. So once again, we have these pathogen-derived molecules, or these PAMPs, on the outside of the cell. Once those then bind to the pattern recognition receptor, or the receptor here, it sets off that signaling cascade. And then, as I mentioned, NFkB is generally held in the cytoplasm by this inhibitor protein. So once this signaling cascade occurs, it leads to the degradation of IkB, once again releasing NFkB, so that it can function as a transcription factor and move into the nucleus. So as it moves into the nucleus, we're going to have it binding to the DNA, turning on those immune genes, transcribing them into RNA, which then get translated into those proteins and signaling that same immune response. So this, once again, is a very extensive process. There's multiple different signals going on within this pathway. So my project zooms it in completely on looking at the movement of NFkB itself. So as I mentioned, it's either going to be in the cytoplasm in the nucleus. So my project looks at where it is and when it's moving into the nucleus to function as a transcription factor. So the experimental questions that I'm looking at are, do resistant and susceptible strain Bifidobacterium glabrata differ in response to the same PAMPs? And also, which PAMPs are inducing translocation of NFkB, and at what time point is that translocation occurring? So in order to answer these questions, I'll first give a brief overview of my methods and then go zoom in on each of these specific things here. So I start out with some cells. Those cells then get treated with various different PAMPs. And then once treated, those cells are then fixed so that they go through treatment with antibodies to detect where NFkB is. And from there, also use fluorescence in order to then visualize where NFkB is, whether it's in the cytoplasm or the nucleus. So from there, then image the cells and to determine that. So like I mentioned, I started out with some cells. For a while, I was using BS90 and MRI hemocytes. The hemocytes are going to be the blood cells taken directly from the snails themselves. And once again, the BS90 strain is going to be the resistant strain, so it's resistant to that parasitic infection, while the NMRI strain is going to be susceptible to that parasitic infection. And then I've also started using Bifidobacterium glabrata embryonic cell line, or BGE cells. So these cells just provide a unique in vitro model system for the snail itself. They're overall just easier to use, they grow more quickly, and I'm able to get more of the BGE cells onto a microscope slide than I am generally with the hemocytes. So once I have the cells, I then adhere them to a 24-well microscope slide. 24-well microscope slide just makes it easier to do multiple treatments and multiple time points at once, rather than having numerous amounts of slides for one experimental run. So once those cells adhere to the slide, I then treat them with various different PAMPs, and the PAMPs that I've focused on have been peptidoglycan, or PGN, lipopolysaccharide, or LPS, 4-12 myristate 13 acetate, or PMA, and then larval transformation products, or LTP. So once again, that peptidoglycan is going to be representative of the gram-positive bacterial infection, while the LPS is going to be representative of the gram-negative bacterial infection. And then PMA was chosen because it has been shown to induce NFkB translocation in other organisms based on previous studies. And then, so we just wanted to see if it would also induce that same translocation in the snail as well. And then larval transformation products, or LTP, is going to simulate a parasitic infection itself. So these LTP are taken from the Myristidia, so the point in the parasite's life cycle in which it is infectious to the snail. So once these cells have been treated with one of these PAMPs, they then get fixed and go through then a series of antibody binding in order to detect where NFkB is. So the primary antibody is going to be a rabbit anti-NFkB, or an NFkB antibody, meaning it is specifically going to bind to NFkB itself. So these antibodies are the same that we find in the adaptive immune response that I went through earlier. So they are specific to that protein. And then followed by a secondary antibody, which is going to be an Alexaflor 594 anti-rabbit, meaning that it's going to be specific to any rabbit proteins. So looking at the diagram here, we can see that the green triangle would be NFkB, the primary antibody here would be the anti-NFkB, and then the secondary would be an anti-rabbit. And then the most important part of the secondary antibody is going to be this fluorescent tag here. So this is attached to the secondary antibody, which then allows me to visualize where that antibody is, showing where NFkB is. And then from there, I also incubated in a hook stain. So this hook stain binds to the DNA, and this shows me where the nucleus is. So then depending on where this fluorescent tag is, if it's going to be overlaid with the hook stain, then that shows that NFkB is in the nucleus. Versus if the fluorescent tag was outside of the area that the hook stain is, then that means NFkB is still in the cytoplasm. So to understand how those fluorescents work, and then we can look at how fluorescence works here. So there's electrons that start at a ground state, and then at a certain wavelength, they're going to absorb light, exciting them to this excited state here. And then as they return back down to the ground state, they're going to emit a photon or emit light, which allows us to visualize where that's occurring. And this occurs at different wavelengths, so we can look at the graph here with wavelength on the x-axis and normalized intensity on the y-axis. And then, so this excitation, or the absorbance here, is going to be representative of the blue peaks for both the hook stain and the Alexaflor. And then the red fluorescence, or where that photon's being emitted, is going to be from these red peaks. And it's important to note that the hooks and the Alexaflor do not overlap, because since I am looking to see where those lights are, where that light is being emitted, we don't want them both emitting light at the same wavelength. So before going into my results image, I just kind of want to give an overview of what you'll be looking at and what these fluorescents look like in my images. So the hook stain is going to be this blue fluorescence here, and this is showing where the nucleus is. So based on these white images, you can kind of see the outline of the cell is larger than the blue outline itself, showing that the hook stain is only binding to the nucleus. And then the red fluorescence in the middle here is going to be the Alexaflor, showing where NFkB is. And then the best way to determine whether or not those two fluorescents are overlapping is we can directly overlay the two fluorescents as we see here. So this is going to be both the hook stain and the Alexaflor. And we can see here that for the most part the nuclei are completely blue, indicating that NFkB is not in the nucleus and is still in the cytoplasm. And then also one thing to note about each of these images is that they do have a micron scale at the bottom right, indicating how large each of these cells are. And so the difference between then these control images versus the treated images, we can see that once again we have the nucleus is going to be the blue fluorescence, and then the NFkB is going to be the red fluorescence. And then, however, once NFkB has translocated and we overlay the two fluorescents, we can see a little bit of pink where the nucleus is occurring. So this means that there's an overlap between the red and the blue fluorescence. So once again, before going into my results, the experimental questions I'm looking at are: do resistant and susceptible strains differ in response to the same PAMPs? Which PAMPs are inducing translocation of NFkB, and at which time points is that occurring? So the first cells that I looked at are the resistant strain hemocytes, or the BS90 strain, and these pictures are taken in incubation with peptidoglycan, so that's going to be that gram-positive bacterial infection. And we can see in the control images the blue nucleus here and then the red outside of it, indicating that the NFkB is in the cytoplasm and not in the nucleus. However, after 15 minutes, we do see an increase of pink in the center there where the nucleus is, showing that NFkB is functioning as a transcription factor. It's binding to that DNA and initiating that transcription. And then interestingly, by 45 minutes, we do get a decrease of that overlap. So at this point, it appears as if NFkB has returned back to the cytoplasm and is no longer inducing that transcription. So then next, still looking at the resistant strain hemocytes, we looked at LPS, so the gram-negative bacterial infection. Red's a little hard to see here, but so the blue is going to be the nucleus there, and then the red is outside of the nucleus, indicating NFkB is still in the cytoplasm. And then while we don't see as much overlap with the LPS as we did in the peptidoglycan, there is still evidence of some pink here and here, indicating that NFkB is still moving into the nucleus and binding to the DNA. And then similarly, as we did with the peptidoglycan, after 45 minutes, the nucleus is free of NFkB, and NFkB is back in the cytoplasm. So the results for just the resistant strain hemocytes are that both peptidoglycan and LPS, we can see translocation occurring at that 15-minute time point, but once again, then not at that 45 minutes. So from this, we conclude then that gram-positive and gram-negative bacteria are inducing the NFkB pathway in this resistant strain as an immune response within the snail. So next, I looked at the susceptible strain hemocytes, and so once again, in treatment with peptidoglycan, or the gram-positive bacteria, we have the blue nucleus and then the red NFkB on the outside. And so these were shown to be slightly different from the resistant strain, and that we do see blue nucleus all the way across the board at each different time point. So this is showing that translocation is not occurring in treatment to peptidoglycan in these susceptible strain. And then similarly, we see the same thing in treatment to LPS, or the gram-negative bacteria. So we have the blue nucleus and then the red NFkB, and once again, that's occurring all the way across. They all look very similar, and that NFkB is not translocating to the nucleus, and it's still remaining in the cytoplasm. So the results for the susceptible strain is just that PGN, or peptidoglycan, and LPS, neither of them are causing this translocation to occur. So we conclude then that gram-positive and gram-negative bacteria are not inducing that NFkB pathway, and that is not functioning as part of their immune response in those susceptible strains. So then next I looked at the BG cells, which is that embryonic cell line. And this once again is then in treatment to peptidoglycan. And so we can see the blue nucleus followed by the red on the outside. And however, at 15 minutes we do see this pink nucleus again, indicating that there is overlap between the red and the blue, showing that NFkB has translocated into the nucleus at this 15-minute time point. But then once again, by 45 minutes, we do not see that translocation occurring anymore. So then next, I looked at PMA with the BG cells. And so this appeared to not induce translocation. We can see that the cells look essentially the same at the control 15 and 45-minute time points. However, one thought we had was that PMA is known to readily cross the cell membrane, so it is possible that this translocation could be occurring before the 15-minute mark. And then the third treatment I looked at for the BGE cells was the LTP, or those larval transformation products representing the parasitic infection. And so at the control, once again, we see the blue is the nucleus, and then the red slightly on the outside representing the NFkB. And while we do not see any translocation occurring at 15 or 45 minutes, we do have these weird red rings forming around the cells. So while the LTP is not inducing NFkB translocation, in this time point, it is causing some sort of reaction from the cell, causing it to form these rings outside of it. And so we're not entirely sure what that's from or what that reaction is. And then we do see a decrease. There's a few rings here, but not as much as we see at the 15 minutes. And so the BGE results just show that peptidoglycan did induce translocation of NFkB, once again within that 15-minute time point, but not by the 45-minute. However, there's no translocation evident when treated with PMA or LTP. Some future questions to look at could be for PMA is whether or not that translocation is occurring before 15 minutes, and then with the LTP, what is causing the rings to form around the outside of the cell. So in conclusion for the BGE results, we can conclude that only gram-positive bacteria are inducing that NFkB pathway as an immune response in the embryonic cell line. So overall, the study can conclude that gram-positive bacteria does in fact trigger the NFkB signaling pathway in both resistant strain hemocytes and BGE cells, however not in the susceptible strain hemocytes. And then similarly, gram-negative bacteria is triggering that NFkB pathway in the resistant strain hemocytes, but not the susceptible strain. And then neither PMA or LTP appear to be triggering that same pathway. So from these conclusions, it's interesting that we do see that this NFkB pathway is occurring in the resistant strain for both gram-positive and gram-negative bacteria, but we do not see that in the susceptible strain. So this does show that there is a difference in the immune response between those two strains. And so one future way we could look into would be why this isn't occurring in the susceptible strain. At what point in the NFkB pathway is there a malfunction preventing NFkB from translocating from the cytoplasm to the nucleus and turning those immune response genes on? And so finally, I'd like to end by saying thank you to Judith Humphreys for advising me throughout this research project. Thank you to Dr. Dave for advising me throughout this presentation. And then thank you to Wayne Krieger and Joy and Stan for providing all the materials to do this research project. And then references and any questions.
Where you see the NFkB on the outside?
That's what it, because that's what.
Do you think it's traveling to like the membrane? Like what part?
It could be. I mean, that's what it kind of looks like, but not entirely sure. Yeah. Because obviously, yeah, the red is demonstrating where NFkB is, so it is showing that that's where it is, but I'm not sure why it's going there instead of to the nucleus or just staying in the cytoplasm.
So what else are implications of those results?
One implication would just be that the resistant strain is using that NFkB pathway, and that could be functioning as to why that strain's resistant in comparison to the susceptible strain. So it could be that NFkB is helping to make it a more resistant strain than the susceptible ones.
Do you think NFkB might function in the immune system and be vibrato based on what you saw?
I would say yes. I would say it is functioning to activate that immune response and turn on those genes.
So is the snail genome sequenced?
Yes, it is.
And so have you taken a look at, I know you didn't tell us all of the components of the signaling pathway for NFkB, but have you had an opportunity to take a look and compare susceptible and resistant genes upstream of NFkB, but downstream of the receptors to see if there's a difference to explain why the susceptible ones are?
Right. I specifically have not, but I know other students in Judith have that.
The upstream components are there, but the genome was sequenced from a new strain that they went to Brazil to bring back. So we can't use it to directly compare resistant and susceptible, but we found in that genome the components upstream from TLRs down to NFkB.
So it's two different receptors for for peptidoglycan LPS. So that would mean that it's some place where those two signal transduction pathways meet.
And we have
That's-
peptidoglycan recognition receptors. And beta-glucan, I haven't seen anything on a specific LPS, but it could be one of the TLRs.
Other questions for Andrea? Okay, well, thank you very much.